We continue on in Chapter 9, Genetically Modified Organisms. We've already covered the topic of gene expression, and now we're going to cover the topic of recombinant DNA technology. In this discussion, we are going to highlight bacterial plasmids and how these are ideal vectors for the transfer of genetic material. Also, to talk about restriction enzymes, ligases, and lastly, we'll discuss an overview of the entire cloning process. Before we begin talking about plasmids, we need to highlight the ways that bacteria can transfer genetic information. The first mechanism by which bacteria can take up foreign DNA is in the process of transformation, and this is when a plasmid vector or a segment of chromosomal DNA is somehow introduced into a bacterial cell, and I will discuss this process further in a moment. We also have the process of conjugation. This is when there's direct physical contact between two bacterial cells. One bacteria will act as the donor and transfer genetic material to the recipient bacterium. Thirdly, the process of transduction is when a virus infects a bacteria. It will then transfer genetic material from that bacteria to another bacteria. So going through those a little bit more slowly, as I mentioned, bacterial transformation, you can either take up fragments of DNA or you can take up a plasmid. When you take up short fragments of DNA from the surrounding environment, the bacteria can either integrate that DNA into its chromosome, as shown in the lower left-hand portion of this figure, or what will happen is the DNA will be recognized as foreign and will be degraded. The other possibility is that an entire plasmid can get taken up by the bacteria, and this will not usually integrate into the chromosome, but will rather be maintained independently as well. Conjugation, as I mentioned, is direct contact between two bacteria, one acting as the donor and the other as the recipient. So the donor has something called a pilus, and the pilus acts as a bridge between the two bacteria. It is a means by which the other bacteria is pulled towards the donor bacteria. They then can transfer genetic information going from the donor to the recipient. Upon completion of that process, we know that the recipient bacteria can now act as a donor as well. And that is shown here with this electron micrograph. You can see that the bacteria on the left is the donor, the recipient is the bacteria on the right. You can see that pilus acting as a bridge between the two, and what will allow for the two bacteria to be drawn towards one another. The final method by which bacterial DNA can take up foreign DNA is by viral particles, also known as bacteriophage. These viruses will dock on the outside of the bacteria and inject their DNA into the bacterium. They then degrade the bacterial DNA, as shown here in the purple, gets completely degraded. They then take over the bacterium and use it as a means of generating thousands of new viral particles. They take up the DNA, and sometimes accidentally, some of these viral particles will take up some of the bacteria's DNA that got degraded. So for that type of phage that now is actually carrying bacterial DNA, when it goes and infects a new bacterium, as shown in step 4, rather than injecting viral DNA, which is shown here in this picture as the green DNA, it's injecting the purple DNA, which is another bacteria. That bacteria DNA can sometimes get integrated into the host chromosome, and the new bacteria will now express its own DNA as well as bacterial DNA. But this was all transferred via a virus. So these are the three methods by which bacteria can take up DNA. As I mentioned, in the process of transformation as well as conjugation, oftentimes what's being transferred are plasmids, which are just short, circular pieces of DNA in the bacteria. Well, how were plasmids initially identified? In the 50s, there was an outbreak of dysentery, and they were trying to determine what was the cause of this outbreak. They were able to isolate a strain of bacteria known as Shigella. It's very similar to E. coli. It was resistant to actually 3 types of antibiotics. In 1960, it was determined that plasmids from the Shigella bacteria were isolated with 3 antibiotic resistance genes. So it was not actually the Shigella bacteria's chromosome, huge circular chromosome, but it was DNA on the plasmids that was causing this antibiotic resistance. So they concluded that transformation of bacteria with a plasmid can allow bacteria to naturally acquire new DNA sequences and new characteristics. So by the early 1970s, Stanley Cohn, who was working on plasmids, was the first to recognize that plasmid DNA could be harnessed as a means of transporting genetic material. If one could somehow use the plasmids to carry in the foreign DNA into a bacterial cell, then you had a wonderful vehicle for transferring genetic information. So how is it that we had these antibiotic resistance genes found in the Shigella bacterium? Well, if you have a bacteria that is sensitive to a particular antibiotic, in this case tetracycline, we know that this particular bacteria has no plasmids. However, if we can somehow introduce into this bacterium a plasmid, a small circular piece of DNA that has the tetracycline resistance gene, and we can get that plasmid into the bacteria by the process of transformation, we can now generate a bacteria that is resistant to tetracycline. So if you introduce tetracycline into the media, this bacteria will now survive rather than die. And what we know is that as the cell undergoes cell division, which for most bacteria happens every 20 minutes, we can generate a whole population of bacteria that are tetracycline resistant. So what are some of the key characteristics of plasmids that make them ideal vehicles for transferring genetic information? Well, they're very small and they're circular. They're very easy to handle. They contain just a few genes, and they're not essential for bacterial growth. They are able to replicate autonomously or independently from the host. So it looks like we have this ideal vehicle, but Cohn had a pressing problem. His issue was, how do you actually get the DNA of interest into the plasmid? So going back to our model of the transduction method, in which a virus injects its own DNA into the bacteria, in the 50s it had been observed that sometimes the bacteria was actually able to destroy the virus as opposed to being destroyed by the virus. And we called this process restriction. It could restrict the phage's replication. In the 1960s, several groups were able to determine that this protection was due to enzymes in the bacteria that were able to cut up the viral genome. These were called endonucleases because they cut DNA from within the DNA molecule. The bacterial DNA itself is protected from its enzymatic activity because the bacterial DNA is heavily methylated. These restriction enzymes act like scissors, cutting DNA at specific sequences. The restriction enzymes are basically a defense mechanism used by the bacteria to cut any foreign DNA. If this is plant or viral or human or animal DNA, it will cut the DNA. A particular restriction enzyme will only cut at a particular sequence, and these sequences are always palindromes. So as shown here for EcoR1, it always cuts at GAATTC, and the same is shown on the bottom strand. And again, the reason that it doesn't cut its own DNA is because bacterial DNA is heavily methylated, shown here by the CH3 group. And if the CH3 group is there, then the restriction enzyme will not cut. And so this is true for bacterial DNA, which is heavily methylated. And here's showing a whole slew of different restriction enzymes, and there are thousands of these. But they all cut at various palindromic sequences. Sometimes they generate these overhangs, and sometimes they have a blunt cut, as shown here with the last restriction enzyme. The ALU1 enzyme has a blunt cut. So, as I had mentioned, Stanley Cohn had really examined the plasmids, but it was his work with Herbert Boyer, who was an expert on restriction enzymes, that led to DNA biotechnology as we know it today. So what Boyer and Cohn did is they modified a plasmid. This plasmid is called plasmid Stanley Cohn 1. And what we have are several key features in this plasmid. So this is a bacterial plasmid, but it includes a restriction enzyme, which was Boyer's contribution. It also includes an origin of replication, so that this plasmid can undergo replication when the bacterial cell undergoes replication. And it also has an antibiotic resistance gene here, shown as tetracycline. So if this bacteria is able to be taken up by a bacteria, the bacteria will now be resistant to the antibiotic tetracycline. So we now have a vehicle to get our DNA of interest in the cell. We now have a way of cutting our DNA of interest to get it into a plasmid. But the last piece is that we need a means by which to glue the DNA of interest into the plasmid specifically, and that final step is done with the ligase, which is the same ligase that we discussed in the process of DNA replication. So what are the key components that we need to actually clone a gene of interest? Well, we need a plasmid, we need a gene, we also need restriction enzymes that are going to allow us to cut the gene as well as the plasmid, and we need that ligase, as I mentioned. So Boyer and Cohn, they worked on that first plasmid, and they decided to come up with an instrument. So they used their plasmid that they generated, and they also generated a second. So plasmid 1, as I shown you, is here on the left, and then they generated a second plasmid. They cut both of these plasmids with EcoR1 and joined the two together and made a huge circular plasmid. So it looks something like this. So they took the plasmid Stanley Cohn 1 and they mixed it with plasmid Stanley Cohn 102. The first plasmid, as I mentioned, had the tetracycline resistance gene. The second plasmid had a different antibiotic resistance gene called kanamycin. So they cut the two plasmids, both with EcoR1, completely opening it up, going from a circular fragment into a linear fragment. They then allowed the ends to combine, the sticky ends, and they sealed it up with ligase, and it generated this huge single plasmid. So now that they've pieced together this huge plasmid, they can begin to do additional studies testing the plasmid. So my question to you is, bacterial cells transformed with each of these plasmids will survive under which conditions? So if you're taking a look at the upper leftmost plasmid, PSC101, we know that it's tetracycline resistant. So if you treat the bacteria with tetracycline, it will survive. However, if you treat it with kanamycin, the bacterial cells will die. Or if you treat it with tetracycline and kanamycin, the plasmid and the bacteria will die. If you look at PSC102, it will only survive when the media is treated with kanamycin. However, if you treat bacteria with tetracycline or the tetracycline and kanamycin, they will die. If you're looking at this newly constructed plasmid, PSC101 and PSC102 combination together, you can treat the bacteria with tetracycline and they will survive, or with kanamycin they will survive, or a combination of the two and they will survive. So how did Boyer and Cohn take their plasmid and introduce foreign DNA? Well, that is shown here. They took PSC101 and cut it with EcoR1 as described before, and as you remember, this has the tetracycline resistance gene. Meanwhile, they cut DNA isolated from a frog and they cut that with EcoR1. They then introduced the cut frog DNA with the bacterial plasmid DNA, allowed it to bind, they sealed it up with ligase, and they could then screen for bacteria that only express the tetracycline resistance gene, which means it should also be expressing some frog DNA. What they were then ultimately able to demonstrate is that you could produce frog proteins. Frog DNA was expressed and ultimately translated into proteins, and you could isolate that from bacteria. So the idea is that you could then use these bacteria as mini factories to generate your particular protein of interest. So again, as an overview, what are the key steps involved in cloning a gene? Well, you first of all have to take the DNA of interest and cut that with a restriction enzyme. You cut the plasmid with the same restriction enzyme. You allow for the two to bind, allowing those sticky ends to interact with one another. You seal up the whole new construct with ligase. You then transform that plasmid into the bacteria through the process of transformation. And you're going to select for bacteria which took up that plasmid by using the antibiotic resistance gene to your advantage. You'll ultimately harvest the gene or the protein of interest. So how is this method used today? Very similar ways. You take your DNA and you introduce it into your plasmid. That gets then introduced into the bacteria by transformation. Ultimately, you allow for that gene to be expressed. Either you can harvest the gene at that point, or you can allow the gene to be ultimately translated into protein and you can harvest the protein. And these are used for a vast array of assays and methods. Things such as in forensics, we use it for DNA fingerprinting, gene therapy. We also use the protein for things such as producing vaccines, things like human insulin as well as human growth hormone. So I want to highlight briefly how the production of human insulin involves recombinant DNA technology. We know that there's two types of diabetes. Type 1 diabetes is for patients whose pancreas isn't making adequate amounts of insulin. These patients are said to be insulin deficient. This is in contrast to type 2 diabetics, which are insulin resistant. Their pancreas are able to make the insulin, but the cells in the body are not responding appropriately to the insulin that's made. So this is a situation in which they are not properly using the insulin that's there. Insulin is made in the pancreas, and it's necessary to metabolize carbohydrates or glucose, which is the way that we fuel our cells for energy. Diabetes leads to increased blood glucose levels, and we have an inability to metabolize the glucose in the blood and an inability to store the glycogen in the liver. So for patients who are type 1 diabetics, the food is broken down into glucose. Glucose then enters the bloodstream, which is supposed to trigger the pancreas to release insulin as well. And it's the presence of insulin that triggers other cells to take up the glucose. So what happens is you have a huge increase in blood glucose levels because the glucose is not actually being taken up by any of the cells. For type 2 diabetics, as they consume the food, the various carbohydrates do get broken down into glucose. That ultimately stimulates the pancreas to produce insulin, which it does. However, there seems to be an inefficiency of the cells in the patient's body to actually take up the glucose. And the net result is you have an increase in blood glucose levels in the blood as well. So Boyer, from Boyer and Cohn, that we've talked about extensively, he went on to co-found Genentech, which stands for Genetics, Engineering, and Technology. And one of Genentech's first products was generating human insulin. So again, they used a plasmid. They cut the plasmid with EcoR1. They introduced the gene for human insulin, combined that into the plasmid, introduced that into the bacteria by the process of transformation, and then allowed the protein to be the gene to be expressed, the protein to be isolated. They would collect the insulin made by these bacterial cells, which were acting as little factories for them. And this was happening in the late 70s. And Humulin, which is basically human insulin, was licensed by Genentech and ultimately partnered with Eli Lilly. It was approved by the FDA in 1982 and was the first recombinant drug on the market. It's now used by approximately 4 million people per day. So some key vocabulary that you should know. Recombinant DNA is simply a DNA molecule containing foreign DNA sequences linked to host DNA sequences. A clone is just a molecule, a cell, or an organism that was produced from another single entity. And when we talk about cloning, that's making many copies of the gene. So in summary, you should understand the role of restriction enzymes, the role of vectors such as plasmids and ligase in the process of cloning. You should understand the landmark experiments performed by Boyer and Cohn, and you should know what the major steps are in cloning a gene.